Thank you, Vincent. I'm really pleased to be able to be here today and to start off the new year. So Happy New Year to everyone. And we thought what we would do is spend some time talking about the overall vision, mission, and where we're going with Spark. So as we go through, I wanted to start out by just a conceptualization of why we started out doing Spark in the first place. So many of you know, and I think have compared stories with each other, other families, people in your community, with individuals who carry the diagnosis of autism. And as you do so, you'll realize that there's some things that are similar between your family member and others, and other things that are really distinct or unique or different. And we realize that this is really a broad spectrum as we're doing this, and there are individuals with different both abilities and challenges within the community. But within this group, it was somewhat challenging as we started thinking about developing treatments because of that heterogeneity within the group. So from a scientific point of view, that heterogeneity is challenging because autism really, the way that we currently use that term, is probably not just one condition. In fact, we call it a spectrum. And when we think about either etiologies or challenges or symptoms, again, it's not just one particular type, but probably is several different things or even several hundred different subtypes all within that umbrella. The challenge from a research point of view, and I'll remind people that at this point there is no FDA-proven or FDA rather certified treatment from a medicinal point of view that is for the core symptoms of autism. There are medications that we use to treat anxiety or medications for some other symptoms that can go along with autism, but nothing that treats the core symptoms. As researchers were trying to understand both how the brain is different as well as trying to understand what treatments might work, we would have researchers, for instance, who would take 100 individuals with autism, and they might do brain imaging studies or other behavioral studies, and they get some nice results. And then another researcher would try and what we call replicate or do the same study again, but they'd take 100 different individuals, all again with the same label or diagnosis of autism, and they wouldn't get the same results. And that has been really challenging for the field because we haven't seen the robustness, the reproducibility across studies in the field, and therefore we haven't been able to move the research forward, and also we haven't been able to find treatments that work. So in doing this, this has really been an unmet need in terms of being able to power the research that ultimately powers the new treatment supports for individuals in our community. And so as we conceptualized Spark, it was to be able to make it both, number one, that families could participate in terms of thinking about what the research agenda should be. What is it that we should be doing to support our community? Number two, that we had enough people to be able to power that research going forward and to sort of accelerate the scale of what we were doing in terms of being able to move the needle. So in other words, make it easier for researchers to do what they were really good at doing, partner with them in terms of setting the agenda to a certain extent, as well as being able to help tell them what would be meaningful differences, and then to make it easy for them to do their research by saying that we had volunteers out here to be able to participate in research. So as we've been doing this, as I'll get to later on, this has been actually very successful with Spark. You all have been really incredible in terms of being able to participate in this, and we anticipate doing much more of this as we're going going down the road. So in doing this, as Vincent had talked about originally, one of the dimensions that we thought would be important to stratify or divide the group in were things like, for instance, age. There are different challenges at different age points within the continuum of life, and we realized that there are different questions, therefore, at different ages, so we wanted to have people of a wide variety of ages. We realized that there would be differences even in sex, so that there are some challenges that males have more than females or females more so than males. And we also realized that there might be differences in terms of the genetics. The genetics would provide an important ability for us both to be able to subdivide the different subtypes, but also to get, I call them anchors, but some really insightful knowledge to be able to understand how the brain is working differently from a molecular point of view. And to have evidence that was really, I consider it incontrovertible, really sort of solid, robust evidence that would help identify some targets that we could use in terms of developing treatments, developing drugs, developing ways to support individuals. So as we've done this, we started conceptualizing what the different causes might be for autism. And these are some of our best guesses. I won't say that this is set in stone at this point, but as we think about the different causes or different contributors to autism, let me be clear, even though it will sound sacrilegious because I'm a geneticist, it's not all genetic. There are important genetic factors. We know this based on twin studies, being able to look at identical and non-identical twins, and based on now some of the genetic studies both that Spark and others have done, that there are both inherited factors, so things that get passed down from mom and dad, as well as things that are happen not in terms of being passed down from mom and dad, but what are we call de novo mutations. So things that are new in the individual with autism. Many of those happen either from cells that are what we call gametes, or egg cells or sperm cells that are passed down from the parent to make the new individual. But some of them, we call them somatic, actually are not in that egg or sperm, but happen after the point of fertilization. It's a minority of cases, but we do see that. Importantly, the environment or exposures in particular during either the mother's pregnancy or also shortly after birth are probably important contributing factors, as well as just incidents that happen over the lifetime. So all of these contributing factors. Some of you know that we've had several questionnaires or questions to ask you about environmental exposures. That all goes into being able to understand this better, and we'll be doing more of that in the future as we understand specific things that we should be asking about. So as we've done this, it's been really a massive program going out through the United States. This is just to show that we have 31 clinical sites. Many of you have worked with your local medical centers, worked with your local healthcare team there to be able to join Spark. Some of you have joined not through those centers, but you may have been affiliated with them in some way. You may have been given the opportunity, for instance, to work with them to help collect saliva or be able to help with registration. But to say we've got a great network of really institutions I consider to be top-tier institutions, specifically in autism. As we've done this, we've been very successful. This is showing what recruitment numbers have looked like since we began this, and I'm really thrilled to be able to say that we have now over 200,000 participants in Spark. We're getting closer and closer to having 100,000 individuals with autism. I hope we'll be able to surpass that number relatively soon. And that really has enabled us to create the community of individuals who are research ready to participate, and you have done an amazing job in terms of that, and I'll come back to that later. As we've done it, if you haven't seen it, I'm just showing here a lot of the digital communication modules that we have. Vincent has actually been a key player in terms of developing that, but being able to understand what it is we're doing, why we're doing it, and if you don't get all of your questions answered today, I urge you to look at some of this material because there are great bits of stories from families really about what their experience has been through this journey. So as we do this, as we've done this, I wanted to take just a couple minutes to explain what it is we've been doing, and again, more importantly, where we've been going. So for individuals who have consented to be part of Spark, you've done this online. Some people have had help doing it, but everyone's done this online. And for many people, they've sent in a saliva sample. This is an unabashed plug for those of you who haven't sent in your saliva samples. If you have them at home, if they're in your closet, if they're whatever it is, and pushed into the corner, they're actually good for quite a long time, so don't think of them as being expired. Do look at the date, but many of them actually, even if they were sent to you a year ago, are still fine to use. We use this as a mechanism, again, to collect a DNA sample for the subsequent genetic studies. Again, everyone is not required to do the genetic portions of this, but if you do participate in that, then that does allow us to analyze your genetic information and to give you that information back. Importantly, though, the genetic aspect of this is just one piece of what Spark is doing. So as we're doing this, I want to clarify something that probably is a bit confusing. There are some surveys that we send out from Spark that we consider to be core information that is used by the research community. The research community is then able to take any information that you provide online, with or without genetic information, and we de-identify it so that no one knows who you are, but then researchers around the world can use that information to ask questions about autism, and every time you put in one answer, it may be hundreds of researchers who are looking at those answers to be able to understand autism better. So it's a multiplier effect in terms of what you're doing. In addition to that, we have a program called Research Match. That's different than Core Spark in the sense that it's an invitation to participate in research studies from individuals, again, around the world who might be doing research, but it's their own research. They have a very specific question, and you're helping them to answer that research question. Once you participate within those research match studies, after the investigators have had a chance to look at the data, understand it, those data actually get reposited, put back into Core Spark, so then it's something that, again, the entire research community can use. So every time you answer a question for Spark, every time you do something for this, we believe in recycling, reusing, and being able to get it to the largest number of researchers, again, protecting your identity, but to make sure that your investment pays off at the end of the day. So as we're doing this, I have to admit that when we first started Spark, a little over, I guess, gosh, now it's been 4 years since we started as I think about this. As we started doing this, we weren't sure what the response from the community was going to be. So we didn't actually know how long we would go forward. So we sent out a survey, an online survey, to understand from the community, what would you be interested in, how long would you be interested in participating in Spark? Was this, something that you would be interested in doing potentially for 20 years or longer? And I think we were really, really heartened to see the incredible response from the community. Over half of you, almost 60%, said they'd be willing to participate in Spark for more than 20 years. Some people, for good reasons, said, I don't know, you tell me what the options are. And many people said, at least for the long term, 10 or more years, the majority of individuals were willing to participate. So based on that feedback, and I want to emphasize, we really listened to you in terms of hearing that feedback. Based on that feedback, and due to the generosity of the Simons Foundation, we have a long-term commitment to Spark that really is going to go decades into the future to be able to, again, understand for many individuals how things change for them over the life course, but to take the same person and follow them over their life course and as things change with the ups and downs that we all have, to be able to understand what those differences are, and again, to be able to power for researchers, power this research in the long term. So that's something I want to emphasize. We're not going anywhere. We're staying here for a long, long time. As we're doing this, we also asked you some other questions in terms of the ways you'd like to be able to participate in Spark. And so there are several different ways. As I said, for the things that we do at Spark Central, we do those as online surveys, online things that people can do so that anyone's able to participate. And there was really overwhelming response, both for adults with autism as well as parents of children with autism, that this was actually something they're quite interested, quite able to do and willing to do in terms of this. And so that is something, again, based on that feedback, we'll have a long-term commitment to doing those online types of studies. And we're going to try some creative ways of doing more things online, some things that researchers haven't always traditionally done. We've also been thinking about wearable devices, so things that people could, whether it be something like a Fitbit or an Apple Watch or something that they could have in their own home, but give us some more granular data or some more specific data about how they were feeling, how their bodies were working, how things might be changing, to be able to get some of that information. And again, anyone who would do this would, of course, do this knowing fully what we were collecting and why we were collecting it. And it was incredible. 3/4 of individuals out there said, yes, that would be something I'd be willing to try. And so based on that, we'll take that information, and we are starting to try some different devices. We want to make sure they're going to work before we ask anyone to volunteer to wear any of them or participate in any of them, but we absolutely are going to have a segment going forward in terms of trying to collect data like that. We also asked about, again, things that would be what I'd call research match studies in the sense of studies that we won't be conducting ourselves at Spark Central, but that will be things that are in-person evaluations. We realize that not everyone will necessarily be close to a site where researchers might be doing that, and it's more burdensome. You've got to get, to the place, it takes more time, and so understandably, a few less people, but still, again, the vast majority of individuals willing to do something like that. And so based on that, it's information that our researchers in the research match are certainly now coming more and more to us to be able to match individuals interested in research with researchers doing this with in-person evaluations. And then finally, we started getting down to things that really are, when we think about the risk and the benefit, could potentially be somewhat riskier, and that is clinical trials. So this is trying new medications, hopefully with the purpose of finding something that's going to work for at least some individuals with autism, but they're things that are not proven yet. And so while we certainly hope that they're going to be helpful, we don't know that for sure, and that's why we do research. So a third of individuals, both adults and parents of children, said yes, even for what I consider to be the most the biggest contribution, the heroes out there who are willing to do it, a third of our community willing to do clinical trials. And so because of that, I'll get to it in the end, we'll continue to support clinical trial work in autism as well. So I'm going to start at the very beginning. And for many people in our community, they themselves or their child has autism, but we couldn't necessarily know what their brains were like in terms of very early development, either development in utero or development in early life, because by the time they join Spark, on average, our participants are 8 or 9 years old. So that period of time has already passed, and we can't sort of go back in the clock and be able to study things that have already gone by. But on the other hand, some of our families are still having children, and for those individuals who are having additional bundles of joy adding to their family, there is the opportunity to see what the brains are like in terms of those developing children, in some cases in utero and in some cases shortly after the child is born. So we've called this group of studies that is done on those children baby siblings, because they're the siblings or the brothers and sisters of an individual with autism. By virtue of that, they're at increased risk for autism, not to say they're going to get it, because the majority of them won't, but they're at higher risk than the general population. And so by doing that, we can more efficiently study individuals who might develop autism and then have a nice control group of individuals of people who ultimately don't develop autism. So watch for this, and for those of you, again, where you might find us asking about questions of, are you pregnant, or have you had a new addition to the family? A large part of the reason why we're doing this is to power these types of studies of young infants. As we're doing this, and as I alluded to before, we're also going to continue following the individuals in Spark over time. And that's because we realize that the life course changes. There are new challenges, new opportunities, both new joys as well as new challenges, and we want to be able to capture that. And importantly, I'll say, even though I'm a pediatrician, one of the times when we start planning is even in adolescence, even during teenage years for the transition when our young people become adults. And that's because many of the services that are in place, many of the things that we want for our young people to become more independent, we need to start planning early. In Spark, we have a specific issue that comes up that I wanted to alert you to, which is that when our young people turn 18, from a legal point of view, things change. That is that they then become, from a legal point of view, adults. And when that happens, some of them become independent. And that's a wonderful thing, for them to be able to do things on their own and legally to make decisions for themselves. Because of that, it is important for those individuals, once they turn 18, if they can make that decision to participate in Spark, we want them to be able to, and in fact, legally, we need them to make their own decision about participating in Spark. That is, we need them to consent to do online what you as parents did for them, to give permission to continue following them. We realize not everyone will want to do that, and that's okay. We're not obviously forcing anyone to do that, but we do need to be able to get their permission and sometimes new contact information, a new email address, for instance, to be able to reach them so that they can fill out surveys or answer questions that we might ask. we know that some of our individuals, even though they turn 18 and become adults from an age point of view, a chronological point of view, they're not independent and not capable of providing consent for themselves. And so we need parents in those situations to be able to just tell us that, and that they're the legal guardian and continue to make the decisions for that individual. And then based on that, that parent's consent will allow us to continue working with your family long-term over time. But I do, again, want to emphasize that this is very much a long-term commitment on our part. As we're doing this, we've learned from researchers that there's some information that we haven't yet collected from you. And so based on that feedback, knowing what's important, what are the burning questions in autism that need to be addressed, we've prioritized the additional information we're going to be seeking from you. Part of this is because things change over time, and in particular when it comes to mental health issues, we know some of these do change over the life course, and so we're going to be asking very specific questions about that. We want to be able to know, in some cases, some metrics, some things that you may have gotten testing or your child may have had testing for in school or by a psychologist. And so when it comes to, for instance, things that we call autism diagnoses, formal, what we call an ADOS, which is a test, an evaluation a psychologist does for autism, or being able to get metrics like IQ, that's something that helps us to understand to an approximation at least where someone is along the spectrum and where there might be specific challenges and certain tasks might be easier or harder for certain individuals. In addition, because this is a brain condition, we want to be able to look more carefully at the brain. So both in terms of viewing the brain, visualizing what it looks like with an MRI, for instance, or magnetic resonance imaging of the brain, as well as being able to look at the brain waves themselves. So the electrical activity that flows through the brain through something called an electroencephalogram. We realize you don't all have these in your back pocket or in a binder at home, so we're going to be working with individuals to help us to gather these data, and we probably won't ask everyone to do this because we know this is more challenging, but we'll start it out on a smaller group and expand over time if we figure out a way that works in terms of families being able to contribute this information. As we do this, as I said, we realize that there is more than just, for instance, what your doctor collects in the doctor's office or what your psychologist collects, but information that is happening on a day-to-day basis. And we realize that the way individuals act in different environments is different. So the way someone might act in a doctor's office might be very, very different from the way they act at home. And we want to be able to capture how people are acting at home or at school or in the community because that's where they are most of the time and that's what matters. But we may have to have different ways of capturing that information. So as I said, whether it's by a wearable device like a watch type of device, whether it's something on the phone, whether we can capture, some people have challenges in terms of sleeping. So we've been thinking about how to collect information with when people sleep and how they sleep and if they get a good night's sleep. But we're working with a lot of different technology people to try and develop ways of doing this. We're also going to be experimenting with different ways to ask you questions, ways that hopefully not only are clearer but less burdensome. And so you may see some questionnaires coming from us that hopefully are not quite as long and not as difficult, but may come in what I call bite-sized bits. That is being able to get things that you can do more quickly and don't take as much of your time. So in addition to collecting all of this information, I also wanted to give you an update that we've been very hard at work on the genetic dimension. Again, not everyone has participated in the genetics, and that's fine. But for those of you who have, we've been working very hard at being able to collect that information. I want to emphasize that for most individuals who sent in their saliva samples, we've been able to proceed through generating what we call sequence data, DNA data that we can read out through a couple different platforms, what we call genome sequencing, exome sequencing, microarray genotyping, for those of you who are aficionados. I will say there are a few of you out there where your samples didn't work. So about 10% of the samples that get sent in to us, for one reason or another, those saliva samples haven't worked. So I urge you, if we sent you a message, sent you an additional kit to be able to provide a second sample, it's real important that you do that because without that additional sample, unfortunately, the sample wasn't able to be used. And sometimes that's because there wasn't enough of the DNA in the sample. I won't say that I know all the reasons, but at the end of the day, we need another sample from you. If you need help with that in any way, again, we've got videos online. If you need help with the saliva assistance, we've got online to be able to help with that too. As we've been doing this, we've been sequencing literally thousands, tens of thousands even at this point of individuals to understand what the genetic bases are, and I can say that we've made quite a bit of progress as we've been doing this. As we've started out at the beginning, we were better able to interpret specifically the genetic information from families where we had both the mother, the father, and the child with autism available to us. And that was largely because having mom and dad available allowed us to be able to identify very quickly genetic events that were new in the child and not present in either one of the parents. We've gone beyond that, not just to sequence what we call the trios or the individuals who have families, full families with biological mom and dad, but we've also been looking at many other family structures as well. And we realize that in many cases, we simply don't have access to mother and father for whatever reason. And so we're developing methods to analyze those data as well. It's been remarkable to watch how quickly we've learned, and that's simply because the numbers have been really large. At this point, we've got data on over 7,500 individuals with autism, as well as their family members, and we have almost doubled the number of genes that we know of for autism. And as we understand that information, we're able to provide that back to you. So we've been going through this process and accelerating how much and how quickly we can return the information back. At this point, we're close to 200 families that we've returned results to. I can say I've had the pleasure myself of talking with many of you, as well as we've had genetic counselors, and some families have had their own doctors or genetic counselors at their own institutions provide that information back to them. As you can see, we've returned information to people across the entire age spectrum, children and adults, because most of our participants in SPARK are children. That's why you're seeing most of these results going back to children or families with children, but really we've returned them to individuals across the age spectrum. As we're doing this and as we learn more, we will continue learning more so that if we don't have a result for you yet, we may have a result in the future. And again, if you're able to go to your dashboard, you can see where your samples are in terms of that process of submitting a sample, generating the sequence data, analyzing it. And so if you have any questions about whether or not your sample's been analyzed, you'll see that there. In addition, we send out an email once we've analyzed the sample. If we don't have an answer yet, we tell you that we don't have an answer yet, but we're still working on things. So stay tuned for those of you who we haven't contacted yet. I also wanted to say what happens after that, because it doesn't just end with getting a diagnosis or getting genetic information. Really, it's just, to me, the beginning of the next chapter. And so we have a sister study called Simon's Searchlight. It's also in the past been called Simon's VIP, because these are very important people. But everyone who's participating is important, and Simon's Searchlight is just one segment of the community that then has genetic information, and we try and create a sub-community. So other families that also have the same gene that's involved, allow them a place to be able to share information, learn from each other, and then also to be able to learn more deeply about that condition and connect you with researchers who are studying that particular condition. So as we do that, this is a pretty long process. What I'm trying to show here in what I call treatment development is what it takes, at least for genetic conditions, rare genetic conditions specifically, to go from the original step number 1 of identifying a gene all the way to step 8 or even 9 of beyond clinical trials, having medications or treatments or strategies that work. It takes lots of steps in the middle to understand the condition, how it changes over time, to be able to have tools that researchers can use to do research, which include everything from cells from people with these conditions to animal models that we make, to understanding how the disease happens, how the condition happens, how molecules in our brain act differently, to start to develop, before we do it in humans, new strategies, new treatments, and try those out in a dish in the laboratory or potentially in animal models. All of this ultimately to test it in people and hopefully find things that work. And this whole process, as I said, is many different steps. At the Simons Foundation, though, we've been very committed, not just, again, to stop at step one, but to continue going through all of those steps as we go to crossing the finish line in terms of developing new treatments. And so for many of these conditions, we've either funded research from researchers working on these, helped to make many of these reagents, these cell lines, these animal models, and in fact, working with partners to think about the best strategies for each of these genetic conditions. So with that, to realize that this is a pretty large task, this is an example list of the different genes as well as what we call copy number variants or particular segments of chromosomes that are related to autism that are part of Simon's Searchlight. And as we're doing this, we're working with many patient advocacy groups, many foundations who are working on this as well to partner with them to be able to, again, bring more resources to the autism community. As an example of how many people we have in some of these communities are more mature because we've known about these conditions for a longer period of time. So those are some of the larger bar graphs on the left. Some of the bars and the genes that are along the right are relatively newer conditions, but many conditions at this point I've had the pleasure of seeing get over I think of 100 as being a big milestone. In terms of when you get to 100, there are it's really sort of a mature community and a lot of people paying more attention to that. So we try and help families, groups to be able to increase their numbers. And this particular portion, Simon's Searchlight, is something that we have internationally. Families from around the world are able to participate. As we've done this, in some cases we've been able to host or at least participate in family meetings around the United States especially, but sometimes around the world, and having families come together. And I'm not meaning to focus on the details here, but to see that at each one of these family meetings, it's an opportunity for families, sometimes for the first time in their lives, to meet face-to-face and meet other families with those conditions. And as we do that, in many cases, we're able to bring research teams to those family meetings so that families can participate in research in a way that's easier and more accessible to them. And then, again, as part of the spirit we have in the community, to have those researchers present that information back to families so that we can all learn about this as quickly as possible. As we're doing that, and as we're making advances, as I said, analyzing your information just one time is not enough. This is simply a graph to show that in some cases we've analyzed data like exomes or genomes, and about a quarter of the time in this particular study, and this, I'll also say this was not an autism study, but a quarter of the time an answer was found, but about 75% of the time an answer was not found until researchers went back 3 years later and looked at the same information again and realized that about half of those cases they couldn't figure out 3 years before, they've now been able to figure out. And so again, the field is moving quickly. That's exciting. By moving quickly, we also want to make sure that we keep going back over and over and over again to be able to look and see if there are new diagnoses or new information we can find for you. As we're doing that, I want to assure you that we're using the most advanced technology from a genetic point of view to find the answers. For those of you, again, who are aficionados, in some cases we're doing whole genome sequencing, which means looking at every one of the 3 billion genetic letters to try and find the answer. And we're also empowering the very best and brightest researchers around the world to use this information to help to discover this information as well. So in doing so, I want to make one important point, and this is a really important point that I've had several questions about from many of you. And that is that even though there's a large genetic component to autism, it's not the same in any one person or any different families, but the type of genetic change also is different. So let me try and give you an example. On the left, I'm showing you circumstances in which there might be one strong genetic factor, which is really the main driver or mainly accountable for what happens with individuals with autism. In those cases, those individuals tend to have more challenges simply than behavioral issues in terms of being able to connect socially. Oftentimes, those individuals may have neurological issues, they may have epilepsy, they may have learning disabilities, or even more commonly, intellectual disabilities. And those are oftentimes, right now, at least in the early part of 2020, those are the ones we're able to more readily diagnose and return that information to families. We recognize that. It's easier for us to pick up and to see. On the other hand, there's something that I'm calling on the right in yellow, polygenic risk score. And what I mean by that is at the other extreme, those are genetic factors, but it's not just one factor. You don't see just one sort of big box, but in fact, there are many, many little genetic factors. In some cases, we think probably dozens, if not hundreds, of genetic factors, all in combination that determine someone's risk for autism. Folks that have that type of genetic predisposition to autism oftentimes are the individuals who actually don't have as much in terms of intellectual disability. In fact, many of those individuals are quite gifted, quite intelligent, some of them even geniuses, but have difficulty relating from a social point of view. And so those genetic factors, it is going to take us probably 100,000 individuals with autism to be able to start disarticulating that, understanding what the genetic pieces are. Because of that, we don't have that type of information to return to families at this point. We haven't figured that out, but that's why we originally started out with such a large number, is because individuals who have that type of genetic, we call it architecture, that type of genetic contribution, it's going to take us large numbers to get there. And in the middle are individuals we're increasingly understanding where it's not one or the other of what I described, but in fact a handful of genetic factors inherited oftentimes from both the mother and the father, but just the right combination of those that account for the challenges. And at the end of the day, when we first meet an individual with autism, it's not clear to us sort of the relative contribution of each of these types of genetic factors, and that's what sometimes feels like it takes such a long time, because we're trying to tease apart all of those pieces. So in the last part, I want to introduce this idea of the research match, and again, many of you have already participated, so thank you for those of you who have. But as we did this, we've now had about 2 years of experience under our belt for doing this, and we have a much better sense of how to make this work efficiently and effectively for you as well as for the research community. So as we've done this, as I said, we have some of these where you're invited to come into a particular study center. Oftentimes it's a medical school or a medical institution. Other times, though, and in fact a little bit, about more than half of ours are actually online surveys or things that you can do from home. As we're doing this again, all of this is, at least the online portions of this are de-identified, so you don't have to worry about your identity being out there. But of course, if you're doing something in person, someone's going to know who you are. When you deposit those data, though, all of those data that are deposited are de-identified. As we've done this, we've had about 50% of our families who've been invited to one or more of these research studies, these research match studies, that is, to be able to participate in these studies. We have a process to decide that these studies are worthy of your time and your energy. So we have a panel of both scientists and members from the autism community who review the applications for these studies with us. They vet them to make sure that there's something that is done that's both scientifically meritorious, but also to make sure from a family's point of view that we're doing this in the way that's most meaningful to you, as well as easing the burden. And so I hope in some cases we've had to do this, improve these as the applications come in, to make refinements and to improve them, but it's been great to have input from the community to make sure that we did it in the best way possible. As we've done this, as I said, we've had several different studies. I'm not going to go through each one of them with you, but as we've done this, tens of thousands of individuals who have participated. I'm very pleased to say that we also, at the end of each of these, at least for our online surveys, ask you for feedback in terms of how this was. And out of a 5-point scale, with 5 being a good score, our average score across the online surveys has been 4.5. So I would say a solid A in terms of the feedback we've gotten from you. We also appreciate it when you write in at the end of those surveys to be able to tell us either things that were clunky or questions that didn't make sense, or things that you're concerned about might not be exactly the information that the researchers want. So it's been helpful, again, to iterate and improve on that. We've had our first publication that's come out of this, and as we do this, our commitment to you as participants is that we make researchers actually tell us the results of those studies, and we present them either as webinars to you or as what we call snapshots, that is making the information that the researchers have learned available back to you so you can see what it is that your time went into. So, and we'll keep doing that. That's a commitment from us in the long term. So with the research match, as I said, one of the things we're trying to do is to manage the burden on you. Some of you have been just so generous. You've participated in not just 1 or 2, but even 3 or more research match studies. We try and not burden people too much in terms of going to the same people over and over again, but spreading out the opportunities and spreading out the burden. But certainly if you're anxious to participate, we will keep giving you invitations to more and more studies. And I have to say that more and more researchers have come to us wanting to participate in Research Match, so I am certain we're going to have many more opportunities. For those of you who haven't yet had the opportunity to participate in the research, hold on. Things are coming into your inbox relatively shortly, both for individuals who are adults and children, and individuals who may need a parent to help do these, as well as people who can do these on their own. So as we're doing this, ultimately we're doing all of this energy is coming into trying to provide better supports. As we're thinking about it, I'm really quite excited that simply from a technological point of view, when I think about what my phone can do today as opposed to what my phone could do 20 years ago, it's night and day. And that technology in terms of supporting individuals with autism is incredible, whether it's in your phone, whether it's with other wearable devices, whether it's in ways of supporting independence. There's just a lot that I'm optimistic about in terms of technology being able to provide support. In addition to that, there are educational strategies that we're realizing are working much better. For individuals, for instance, who have difficulty with verbalizing, there are many more ways of communication that are proving to be effective, and educational strategies we can use to help make sure that individuals who have a lot going on in their heads can be able to communicate the full richness of that information out to us. So again, something I'm very excited about. But in addition to that, we realize that there are some challenges that probably need some help in terms of medication. And that's been sort of the traditional way that medical scientists have thought about things, and also ways that we want to contribute to the community. I'm proud to say that we have a series now of clinical trials with a medication that we at the Simons Foundation have been sponsoring. This medication is arbaclofen, and we have trials now that are going on in Canada, across Europe, as well as in the United States to test this new medication to see if it's going to be effective for individuals with autism, and even starting to look at some specific genetic subsets. And so this is a stay tuned. I don't yet have the answer in terms of whether or not this is effective. So far the trials are going well, and people are tolerating the medication, so they're not having any problems with the medication. And hopefully within the next couple of years we'll have information to know whether or not these medications are working. In addition to medicines or things that you would put in your mouth, there are also some incredible new gene therapies or molecularly based treatments. And although this condition I'm showing you here, spinal muscular atrophy, is not autism, and I'll be very clear about that, I realize this is different, it is incredible that this condition, which I'm going to use the past tense, used to be the most common genetic cause of death for infants less than a year of age, is now very treatable with two different types of medication, one shown here, Spinraza, and another which is gene therapy for spinal muscular atrophy. This condition used to be fatal for children that had the type 1 form of this disease. And in the next slide, I'll show you, this is a little girl whose permission I have to show you, but this is a picture of her after she was diagnosed with SMA and then treated effectively with one of these treatments. And when she would have been otherwise expected to be dead by 14 months of age, she is in fact a completely healthy, strong, normal child. And by using the learnings from other genetic conditions that aren't necessarily autism, but share genetically some of the same mechanisms or some of the same ideas, we are going to be beg, borrowing, and stealing, but being able to take all those lessons learned and apply them now to all of those genetic forms of autism that we've been talking about and learning about. So to wrap things up, this is a very long-term vision. All of it won't happen overnight, but I want the community to understand how strong our commitment is for the long term in terms of all the different steps that it's going to take. We hope that by understanding the genetic basis for autism, we're going to understand the molecules or the targets we might think about in terms of interventions. Some of them will be directly targeting the gene with gene therapy or molecular mechanisms, other times perhaps with medications, other types with educational strategies or technological supports. As we do this, we really want to speed things up. We're sort of getting impatient in terms of being able to provide more answers for our young people, either our children or our adults, but being able to give them opportunities to be the best that they can be. So as we're doing this, we do need the community to be able to stick together. We appreciate every contribution that's been made, be it big or small in terms of doing this, but we are, again, here for the long term to watch what happens over the life course for your family, to be able to help you through those different hurdles at different lifespans, but importantly, to be able to make sure that you and your family have the best opportunity to the brightest future that we can all have. So with that, I'll be glad to pause here, and we'll take some questions. And again, as Vincent said, this will be online for anyone else you might want to share this with later.
Okay, so I'm going to take a look at some of the questions. Feel free to send in more. We have some time to answer a few here. Okay, so questions about the average amount of time for a saliva kit to be processed, and if you could kind of go over the return of results process.
Yep. So let me try and set some expectations. So in general at this point, with the information we have right now, about 10% of our families are getting genetic results returned to them. And those are, when I showed you that picture, that's that big blue box I showed you in the lower left-hand corner. Again, not to say that other families won't have results, but those are the results we're able to interpret and understand at this point. The whole process in terms of from when you send in your saliva kit to the point that we analyze the data and have results for return, I will warn you, is taking over a year at this point. And that's in part because we bundle things together. So as we do the sequencing and the analysis, we don't do it one person by one person, but we do it, for instance, 10,000 people at a time or even more. So we do them on scale and very large scale, and we oftentimes are doing sequencing twice a year. And so depending on when your sample comes in, if it happens to come in just the right time, it may be, sort of relatively faster. And if it happens to come in just at not the opportune time, it may take longer in terms of getting into sequencing and then getting into analysis. I'll emphasize that this is not meant to replace your usual clinical care in terms of your doctors and your medical team. If you have urgent questions and need an urgent diagnosis, please do go to your local genetic providers, your genetic counselors, your genetic doctors. But if we do find an answer, of course, we want you to have the benefit of that information.
So someone has a question about inherited genetic factors. So basically, they want to know if inherited is about 50%, would that mean that about 50% of people with ASD would also have parents with autism? Oh,
so that's a good question. So in terms of the inherited portion of this, even though a genetic factor might be inherited, it doesn't necessarily mean that that parent has autism or has ASD or is on the spectrum. And that's a really, really important point. Number one is these genetic factors that are inherited probably are not acting alone. They're acting as combinations with other genes as well as other things that happen over the life course. And so within that, we find many times, or it's starting to look many times like they're inherited factors, but some inherited from mom, some's inherited from dad, and it's the combination of those together, along with, again, what happens over the life course determining autism. So in those cases, it might not be that mom or dad, either of them have autism, but it's the combination of that together. There are some families where we see it running in families, where we see either multiple children with autism or a parent that has autistic features or has autism. And in those, we do see more of the genetic inherited variants, but that's something that we're just starting to understand. So we haven't really figured that one out completely yet.
Okay. Someone is asking if Spark would ever share raw genetic data that participants could then have on their own.
Right. So very technical question, which is that there's a lot of genetic information that we're collecting, and there are some people who might be particularly savvy about how to use that information, or they might want to go to a service that might be able to interpret that information. At this point, giving what's called the raw data or the sequence data, most people wouldn't know what to do with that. But if there's someone who really has a specific urgent need to be able to get that, take it to another provider, take it to a service to do that, call us in terms of online at Spark. There's an email that you can send to us, contact us for specific details.
So I'm going to generalize with one of the questions here. It was a little specific about someone that has genetic change, but I think what they're trying to ask is there value in still submitting DNA if they already have a known genetic finding?
Okay, very good. So there are two options. For individuals who already have a genetic diagnosis, if they feel like there's something that's still missing, a piece of the puzzle, and I certainly have had patients who have what I call dual diagnoses or more than one diagnosis. If it feels like that to you, then certainly submitting a sample, joining Spark, submitting a sample, saliva sample into Spark, we will start from scratch and we will see if we see that second dual diagnosis that you might not already know about. So that does have some value. If you really feel like you have a genetic diagnosis and it feels like the right diagnosis based on what your doctors have talked about, you're also welcome to just directly join Simon's Searchlight. We're asking some things that are similar, but some things that are different. If your child has that genetic change and autism, and you feel like you'd like to participate in autism-specific research, you can certainly even participate in both. So you can do Spark, you can join Spark, and you can join Simon's Searchlight. Spark, again, is going to focus on autism and things related to autism. Simon's Searchlight is focused more on, from a specific gene and genetic community, that dimension. So you can do all of the above.
And somewhat related to that, does Spark return genetic findings to parents or siblings that don't have an autism diagnosis?
So the first round of what we've been doing is returning genetic information for genes or genetic changes that are related to autism. So the way that we analyze the data is we've got 150 conditions that we are robust and certain are related to autism, and at this point, anyone we find that has those genetic changes comes up in our analysis. As we're doing it, number one, everyone needs to tell us what they'd want to hear back. So if an individual says that yes, they would want that information back, then yes, we give it to them. They can also say that no, I would not like that information back. I might want to be part of Spark and the study and even the genetic portion, but I don't need to know my own information, and that's fine as well.
Okay. Feel free to continue sending in questions. I know that we do get a lot of questions about genetics, so if you have, we have a genetic expert on the line, so please feel free, if you have specific genetic questions, to send them through.
And I will say sometimes it may be a little hard to find on the website, but we also have, I've done another webinar on genetics, and you're certainly welcome to watch that. For some of you who may have some other detailed questions, that may answer some of your questions if we don't get to them this afternoon.
I guess I can ask a question that might be interesting to folks on the line. What's something that you're excited about for this year related to Spark? So we're going to be coming up on our 5th anniversary. As I said, we just passed 4, so it's going to be really exciting to see, number one, just by how long we've been in this and how much we've achieved. I do expect that we're going to have some major, from a genetic point of view, major publications or major information that's going to come out from researchers using the Spark data, and so I think it's going to be a time when you're going to see rapid acceleration about what we know about those genetic findings. But beyond that, like I said, what's exciting to me as well is just the diversity of researchers who are asking so many questions in so many dimensions, be it the clinical trials with our baclofen or other things, but being able to learn in a lot of ways about not just the genes, but about many other things related to autism. So with this, it's just, I do think in a good way, I want people that are listening to this to understand how special they are and how meaningful their contribution has been. Sometimes it feels like when you're filling out that survey at 10 o'clock at night in your pajamas that, what could you be doing? But the power of what you have done has been incredible for the research community. The number of people who were not autism researchers, but were really talented researchers now coming to work on autism because of what you've done has just been, it's great to have that brain trust in terms of being able to redirect them and focus them on areas because they now have questions they can ask and answer. And I think that was hard for many researchers before. So you yourselves have catalyzed this movement in science to be able to do things that are going to be good for you, I hope, in the long run, but the long run being not too long. Okay, one more question here. is Spark looking at genetics as it relates to co-occurring conditions like ADHD, depression, seizures?
Yes. So the way, again, that we primarily look at the data is to look at, we look at everything in terms of discovering new genes, and once we appreciate that genes really are genes related to whatever the condition is, then we decide what to return. But as we're doing this, we are looking at those comorbidities. So we think of it as clustering. That is that, like you said, some people have epilepsy. What are the genes that might be related to epilepsy and autism? Some people have ADHD. What are the genes that might be specific in terms of that way that the brain works? So that's part of the reason why we're coming back to you to get some richer data, some more specific information about either medical conditions or associated educational issues or behavioral differences, because having that level of specificity allows us to ask different questions than just what is the cause of autism or what are the contributing factors, but now the richness and the diversity. And to me, that's a lot of where the day-to-day challenges come in, is dealing with those other associated features. So some of you have known that this is an era of big data. I think we now, in terms of Spark, can pride ourselves at being at that, within that club of large data sets that can now start to ask and answer these questions. And we hope that over time we'll also be able to enrich the data with data that you report, and for those of you who give us permission, even data that can be more easily sort of put into Spark, which is data that comes from the electronic medical record in some cases, or data that you might share that's from your doctors, as I was saying, in MRIs or EEGs. So we're trying to, again, think about how to make it easiest for you, what's important for researchers, and what's going to move the needle.
So this will be the last question. Someone is currently a participant and they've submitted samples from one parent and the child, but not the second parent. So should that person submit for the second parent, and maybe generally, can you talk about that?
Sure, absolutely. So it's never too late. That's one of the messages I want to give you. So either that saliva kit, if you've got that at home, it's never too late either to send that one in, or if you need to request a new one, it's never too late to send in another parent, or in some cases, families will expand and there may be a new child that's born, or a new child who was born before but now has diagnosed with autism. It's never too late to update us with that information. If we have already run the samples, for instance, of, let's say, one parent and a child with autism, but not the second parent, if the second parent sample comes in, we will then run that second parent and redo the analysis now with the complete family. And there could be something sitting there that we didn't recognize as being new in the child because we didn't have that second parent for comparison. And so by bringing that second parent in, we're now better able to interpret the first data that we had originally and perhaps return a result to the family. So, like I said, it's never too late. If we had a sample that didn't work the first time and asked you for a sample, please feel free to send it in. We always double check all our work, and so double checking our work also requires more DNA sometimes, and so sometimes we may be requesting a second sample from you because we've run out of DNA and we need to be able to double check it to make sure that we're right before we return it to you. So please, 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 if we've said that there was a problem, we're doing our best to use what we've given us, but do help us in terms of sending in that replacement sample.
Okay, so that's all the time we have. Thank you so much, Dr. Chung.